Welcome to our deep dive for medical microbiologists. Today, we're going to be talking about Bacillus mycoides.
Okay.
We've got MicroWiki, a scientific paper, and an image. And what's really interesting is B. mycoides has this dual potential as a biofungicide, but also as a pathogen.
Yeah, it really highlights how one microbe can have such different roles depending on the context. So why don't we start with some of the basics? You're probably already familiar with the morphology of B. mycoides, but you know those rhizoid colonies?
Yeah.
Really distinctive.
They look like roots.
Yeah, like roots spreading out.
Those root-like colonies are key for distinguishing it from its close relatives. Some of them a little more infamous.
Right.
B. anthracis, the one that causes anthrax.
Yeah.
B. thuringiensis, which gives us that B toxin for pesticides.
Exactly. Now, B. mycoides is generally considered non-pathogenic to humans, but it's important to remember in microbiology there are always exceptions. It can cause opportunistic infections.
Right.
Particularly in individuals with compromised immune systems. So we're talking about things like wound infections, which is why proper hygiene and infection control are paramount in healthcare settings.
Definitely a good reminder for those of us working in clinical settings. Have you encountered B. mycoides infections in your practice?
Not super common, but it does pop up occasionally.
Okay.
Particularly when we're dealing with complex cases or patients with underlying health conditions. It's interesting that the image we have actually describes B. mycoides as a potential contaminant in clinical specimens.
Right.
Which, kind of reinforces that we need to be vigilant in our diagnostic processes.
Yeah, and speaking of that image, it also mentions that the clinical manifestations can include wound infections.
Yeah.
Which ties back to what you're saying about opportunistic infections.
Right, and that goes back to, its widespread presence in the environment. B. mycoides is found in soil, water, air. It's pretty much everywhere.
Wow.
So the point of encountering it is high, especially considering its ability to form those resilient spores.
And that brings us to an interesting little quirk I noticed. The image describes B. mycoides as appearing gram-negative under the microscope.
Oh, yeah.
Even though it's classified as gram-positive.
Yeah.
That threw me for a loop. What's the explanation for that?
That's the intriguing phenomenon of gram variability.
Okay.
B. mycoides is truly gram-positive. It's got that thick peptidive lichen layer, but sometimes it can stain variably, so it leads to this apparent contradiction.
Interesting.
It's one of those little mysteries that make microbiology so fascinating.
It really is. Okay, let's move on to how we actually cultivate this root-like bacterium in the lab.
So understanding its cultural characteristics is essential for accurate identification, especially when we're differentiating it from other Bacillus species.
According to the image, B. mycoides is a facultative anaerobe, meaning it can thrive with or without oxygen, which makes sense considering its adaptability and widespread distribution.
It's a survivor.
Yeah. And on blood agar, it forms those characteristic creamy-colored opaque colonies.
Right.
With that distinct rhizoid pattern. And then there's beta-hemolysis, those clear zones around the colonies. I always find it fascinating how these seemingly subtle visual cues can give us such valuable diagnostic information.
They're like fingerprints for bacteria.
Yeah.
And in the case of B. mycoides, those cultural characteristics are really key for distinguishing it from other Bacillus species that might appear similar at first glance.
So let's dive a little deeper into the specific characteristics.
Okay.
Both MicroWiki and the image give us some good insights. For example, it's catalase positive, meaning it breaks down hydrogen peroxide.
Yeah, that's a helpful test for differentiating it from certain other bacteria that might be hanging out in the sample.
Okay. It's also oxidase negative, so it lacks cytochrome C oxidase. And of course, we know it's non-modal.
But don't forget those spores. Spore formation is a crucial survival mechanism.
Oh, right.
Allowing it to withstand harsh conditions and persist in the environment. It's one of the reasons why it's so ubiquitous.
Right, those tough spores can hang around until conditions are favorable again. And one more thing, no capsule.
Exactly. The absence of a capsule is another distinguishing feature that helps with identification.
Okay.
So we've got a non-modal, spore-forming, capsule-less bacterium with a penchant for those root-like colonies. It's a unique profile.
Absolutely. All these little pieces of information add up to such a fascinating picture of B. mycoides. But now I want to switch gears and talk about something that really caught my eye. The research on B. mycoides as a potential biofungicide.
Interesting.
MicroWiki mentioned a specific isolate called BMJ that's being investigated.
Yeah, it's a fascinating area of research, huge implications for sustainable agriculture.
Yeah.
BMJ has shown some promising results in controlling fungal diseases in various crops. Sugar beets, potatoes, tomatoes, even pecans.
I was particularly interested by its effectiveness against Pythium and botrytis.
Yeah.
Two major fungal pathogens that can cause a lot of damage. And this has the potential to revolutionize how we protect our food supply.
It could. Imagine if we could just rely on naturally occurring bacteria like B. mycoides to control those plant diseases, instead of having to resort to those harsh chemical fungicides. It's a really compelling vision for a more sustainable, eco-friendly future for agriculture.
It makes you wonder what other hidden talents B. mycoides might have. Could its ability to control those fungal diseases in plants have applications for managing human fungal infections?
That's a great question, and it really speaks to the potential of the microbial world for finding solutions to these challenges.
Yeah.
Perhaps B. mycoides, with its dual nature as both a potential pathogen and this beneficial agent, holds the key to unlocking new possibilities in both agriculture and medicine.
This deep dive's really making me think differently about all those microbes we encounter every day. But before we get too far ahead of ourselves, let's take a step back and revisit those key points we've covered so far.
Sounds good. I think we've laid a good foundation.
Yeah.
But there's a lot more to uncover in the world of B. mycoides.
So stay tuned, because in part 2, we're going to delve even deeper, exploring its genomic features, evolutionary relationships, and more.
Welcome back to our deep dive into Bacillus mycoides. You know, we've talked about its unique characteristics, its duality, beneficial in some contexts, potentially harmful in others. Now let's shift our focus to its genetic makeup and see what secrets we can uncover.
Okay, I'm ready to put on my molecular detective hat. What can the genes of Bacillus mycoides tell us about this fascinating bacterium?
Well, genomic studies have revealed that B. mycoides has a surprisingly diverse gene pool. And this diversity is likely a key factor in its adaptability, allowing it to thrive in so many different environments.
That makes sense, considering we find it in soil, water, even air.
Right.
It's clearly got the tools to handle a wide range of conditions. But how does all this genetic diversity relate to its evolutionary history?
Yeah.
Particularly its relationship to other Bacillus species like B. anthracis and B. thuringiensis.
Genomic analysis actually confirms those close ties we talked about earlier.
Right.
By comparing the DNA sequences of these different species, scientists can actually trace those evolutionary pathways and pinpoint common ancestry.
So it's like building a family tree for bacteria based on their genetic similarities.
Precisely. And while they share a common ancestor, each species has evolved unique genetic adaptations
Okay.
that give them their distinctive traits. So, for example, B. anthracis acquired genes that enable it to produce the anthrax toxin.
Right, making it a dangerous pathogen.
Exactly. And B. thuringiensis has those genes for the bee toxin, which makes it so useful for pest control.
But what about B. mycoides? What unique features have shaped its evolutionary journey?
You know, researchers are still actively exploring that question.
Okay.
But we do know that B. mycoides harbors genes involved in the production of various secondary metabolites. These are compounds that aren't essential for the bacteria's basic survival,
Right.
but they often play a role in its interactions with other organisms.
So could some of these secondary metabolites be involved in its biofungicidal activity?
Possibly.
Maybe that's how it fends off those fungal pathogens.
That's a very plausible hypothesis and an active area of research. Scientists are trying to pinpoint the specific compounds that are responsible for those antifungal effects. Imagine the possibilities if we could harness those compounds for agriculture or even medical applications.
It's amazing what we can learn about a bacterium just by studying its genes. And with all the advancements in sequencing technology, I imagine we'll be able to uncover even more secrets in the future.
Absolutely. Genomics is constantly evolving, opening up new avenues for understanding the microbial world. But for now, let's step back from that microscopic level and think about B. mycoides' role in the broader ecosystem.
You're talking about its ecological role.
Yeah.
I mean, we know it's commonly found in soil, water, air. What exactly is it doing out there?
Well, its prevalence in soil is a big clue.
Okay.
B. mycoides plays a crucial role in nutrient cycling. The breakdown of organic matter and the release of nutrients back into the environment. So it's a vital part of that decomposition process, helping to recycle essential elements like carbon and nitrogen.
So it's like a tiny recycler, keeping the whole ecosystem running smoothly.
Yeah, a very apt description. And remember those resilient spores we discussed earlier?
Yeah.
Well, they actually enhance its survival in those fluctuating environmental conditions, allowing it to persist even when things get tough.
It's like a bacterial survival strategy, lying dormant until conditions are right for growth again.
Exactly.
And its presence in water and air further contributes to its widespread distribution,
Right.
allowing it to colonize new habitats.
Exactly. B. mycoides isn't just a loner hanging out in the soil. It's a well-connected member of that microbial community.
Yeah.
Understanding its ecological interactions is essential if we want to appreciate its role in maintaining a healthy environment.
This is really highlighting that interconnectedness of the microbial world and the broader ecosystem. But I'm also curious about the more practical applications of bee mycoids. We've touched upon its potential as a biofungicide, but are there other ways we can harness its capabilities?
Well, the field of biotechnology is always on the lookout for microbes with special talents.
Right.
And bee mycoids has definitely caught their attention. One promising area is bioremediation, using microbes to clean up environmental pollutants.
So could bee mycoids actually help us address issues like contaminated soil and water?
That's the idea. Certain strains have shown promise in degrading pollutants like hydrocarbons, pesticides, even heavy metals. It's like a microscopic cleanup crew for the environment.
That's incredible. What other tricks does bee mycoids have up its sleeve?
Another exciting area is biofertilizer production.
Okay.
Bee mycoids has this ability to fix atmospheric nitrogen,
Right.
converting it into a form that plants can use. This could be a game changer for sustainable agriculture, reducing our reliance on those synthetic nitrogen fertilizers.
And if I remember correctly, some strains also produce plant growth-promoting hormones,
You got a good memory.
so that further boosts their potential as biofertilizers.
Bee mycoids seems to have a real knack for problem solving. It's a real testament to the incredible diversity and potential of that microbial world.
Absolutely. But while all this is incredibly exciting, I imagine there are also challenges and considerations associated with working with bee mycoids, especially considering its relationship to some pathogenic species.
You're absolutely right. While bee mycoids holds immense promise, we need to approach its applications with caution.
Okay.
Safety is paramount, and that means adhering to strict biosafety practices in the lab and carefully selecting strains for applications like bioremediation or biofertilizer production.
So no accidental release of a potentially harmful strain.
Exactly. We also need to consider scalability. Just because something works in a controlled lab setting doesn't mean it'll translate smoothly to a larger scale. Things like environmental variability, competition with other microbes, cost effectiveness, all these factors need to be taken into account.
So it sounds like there's still a lot of work ahead before we see bee mycoids being widely adopted in these applications.
There is, but the potential benefits are significant, and ongoing research is really paving the way for exciting advancements. But before we get too far into the future of bee mycoids, I think it's time to take a short break.
That sounds like a good plan. We've covered a lot of ground, and there's still more to explore, so stay tuned, listeners, because in the final part of our deep dive, we'll be looking into ethical consideration and the potential risks associated with working with this really intriguing bacterium.
Welcome back to the final part of our deep dive into Bacillus mycoids. You know, we've gone from its basic characteristics and ecological roles to its potential in biotechnology, but I want to focus on something that's been on my mind this whole time.
Okay, what's that? We've talked about the risks associated with bee mycoids, its ability to cause opportunistic infections. But we've also seen how it could be beneficial protecting crops, cleaning up pollutants. It's almost like it has a dual personality, like a Dr. Jekyll and Mr. Hyde of the microbial world.
That's a great analogy, and it really highlights how complex the microbial world is. You know, microbes don't fit into neat categories of good or bad. Their behavior really depends on a lot of factors, the environment, the organisms they interact with.
So how do we navigate this duality? How do we get the benefits of bee mycoids but reduce the risks? It feels like walking a tightrope.
It's a challenge that researchers and policymakers are always facing. And you're right, it requires a careful approach, considering both the rewards and the risks.
What are some of the key strategies?
Well, one crucial aspect is risk assessment.
Okay.
Before we use bee mycoids anywhere, we need to evaluate the potential hazards to human health, to the environment. That means figuring out the risks, how people might be exposed, and what could happen if it's accidentally released.
Sounds pretty comprehensive. So would this risk assessment help us come up with safety measures?
Yes. For example, we could use strains that aren't pathogenic. We could implement strict controls during application and have monitoring systems in place to catch any negative effects early on.
It makes sense to be careful, especially since it's related to some pathogenic species.
Exactly. And transparency is really important too. We need to talk to the public, address any concerns they have honestly, and give them clear information about the benefits and the risks. All of that helps build trust and make sure we're innovating responsibly.
It reminds me that scientific progress isn't just about the science. We have to think about the bigger picture, the ethical implications of what we do.
You got it. Open discussions and collaboration are so important between scientists, policymakers, and the public. That's how we set research priorities and make sure those innovations are in line with what society wants.
Now, even with the most careful risk assessment, unexpected things can happen. So we always need to be monitoring and evaluating, right?
Of course, we need to watch how B. mycoides behaves in the real world, outside the lab, and be ready to adjust our strategies if we have to. That's how we make sure we're maximizing the benefits and minimizing any potential harm.
This has really given us a lot to think about, but before we wrap up, I have one last question. If you could sum up the story of Bacillus mycoides in just a few words, what would they be?
That's a tough one, but I think I would say potential, complexity, responsibility. Bacillus mycoides shows us the incredible potential of the microbial world, the complicated ways we interact with these tiny organisms, and the responsibility we have to use their power wisely.
Those three words really capture our deep dive into Bacillus mycoides: potential, complexity, responsibility. And on that note, thank you to all our listeners for joining us. We hope you've enjoyed exploring this fascinating, multifaceted bacterium with us.
And until next time, keep asking those questions, keep exploring, and keep diving into the incredible world of microbiology.